He kōna ipurangi tēnei nā te reo irirangi o Aotearoa.
Kia ora, nau mai haramai ki te ao hurihanga. Hello and welcome to Our Changing World. Ko Clackin Cannon tēnei. We know that Aotearoa is home to heaps of unique animals, found only here and nowhere else in the world. Unfortunately, many of them are also under threat. This week, we head to the Mackenzie region in the South Island to learn about a freshwater critter that most people have never heard of, and to meet the people who are looking out for it. Independent podcast producer Karthic SS has this story.
We've had a good crisp frost this morning. It's probably -2 or -3 this morning, so a good frost to start with, and then it becomes, a beautiful fine day. So yeah, a magic day to be in the Mackenzie Basin.
I'm in Te Manahuna, Twizel, with Dean Nelson from the Department of Conservation. We are standing next to Fraser Stream, a spring-fed system flowing through a paddock. It's a flat landscape, mostly covered in golden brown tussock, and in the distance we can see rolling hills dusted with snow. We are looking for a New Zealand native freshwater fish that's tiny, rare, and not so well known.
My first trap's just up there. I'll just go and pull them out, and anything that's in, I'll pop them in the bucket.
To look for fish, Dean set out G-minnow traps and left them in the stream overnight.
So they have a funnel on each end which tapers down to a small hole, and the idea of it is that the fish will swim in and they find it hard to get back out. And it's typical of the traps that Maori use for catching tuna and other fish. We put a little plastic container, it's an old film container with a few holes in it, in with a little dab of Vegemite or Marmite as bait. And somewhere along the line someone's decided that that's good bait, it attracts fish. I've never really worked out whether it's Vegemite or Marmite they prefer, but...
With a long green string, Dean pulls a trap out of the stream.
Nothing in the first one.
There are 39 traps in total spaced out in this 150-meter long stream, and Dean checks them all one by one.
And nothing in the second one.
He lifts the third one and looks into it.
Aha. Something flapping around in there. Yeah, we've got a couple of fish.
Dean quickly empties the trap into a bucket of water, transferring 2 small fish, and they immediately start circling. We are looking at tiny fish with light brown patterns, just about 40 millimeters in length, the lowland longjaw galaxias.
If you look really hard, you can see it's got a lower jaw that's longer than the upper jaw. You can see that lower jaw when they slow down. A little bit like a bulldog. We kind of affectionately refer to them as jaws, for short.
There are only 7 fragmented lowland longjaw populations in the Waitaki River, and the numbers are so low they are classified nationally endangered in the New Zealand threat classification system, which means they are at a risk of extinction in the short term. They face various threats from introduced trout to land use changes, so the Department of Conservation, University of Canterbury, Environment Canterbury, and other organizations are collaborating to use a promising tool to protect this little freshwater fish. And Dean has been working to monitor and protect the species in Twizel for the past 2 decades.
They get under your skin a bit and you just get really wound up by them, and so they're my big passion these days.
When you think of native freshwater fish in New Zealand, it's usually whitebait, known to make great fritters when they are in season. Whitebait are actually juveniles of 5 different species of fish, all belonging to a family called Galaxidae. They are named for their golden starry patterns on their bodies that look like a galaxy. Whitebait are also called migratory galaxids, as the adult fish migrate from the rivers out to the coast to spawn.
They spawn down in the estuaries of the rivers, and they spawn during a very high spring tide, lay their eggs in the grass, and then when the next spring tide comes in and inundates the eggs, then they hatch and the fish go out to sea and come back as whitebait.
The lowland longjaw is from the same family as the migratory whitebait species, but there's one major difference. They are homebodies.
These guys spend their whole life in this stream. This is their habitat, so they don't migrate, so hence non-migratory galaxias. We also call them pencil galaxias because they're uniformly very thin and narrow, and they only live about 3 years that we're aware of, so they're not a long-lived species like some of the other galaxid species.
New Zealand is home to 12 species of non-migratory galaxids, and there are 13 indeterminate taxa, groups that are yet to be formally described and confirmed as a species. And all of them have been classified to be at risk or threatened, as most of the populations are fragmented and prone to local extinctions. In Fraser Stream, another non-migratory galaxid shares the habitat with the lowland longjaw, the big-nose galaxias.
Big nose are a similar size, but they have a lot more distinctive blotchy brown colour. You know, once again, they've got a feature big nose. If you look at them on their side-on profile, they've got a slightly elevated nose.
While their big-nosed cousins are found only in the Mackenzie Basin, lowland longjaw galaxias are found in two main regions. One here in the Waitaki River and the other in Kauru River in Otago.
The interesting thing is that initially they thought that the longjaws up here and the ones in the Kauru Kakanui were the same species, but since then they've done genetic work and they've split them into two, and they're still both called lowland longjaws, but the ones up here are called Waitaki River lowland longjaws, so they're treated as separate species. We only know them from 7 very small sites in the Mackenzie Basin and the Aotearoa River. And it's really hard to put a figure on it, but I suspect, you know, if you counted all those sites and were able to count all the fish, you might be looking at 1,500 to 2,000 fish in total. So it's not a big number.
Lowland longjaw prefer to live in cool, spring-fed systems and braided rivers.
There's not a lot of work being done on diet, but from what they've found, mayfly nymphs like really clean, unpolluted water. So, you know, hence, you know, springs are ideal in terms of that food and they're ideal for the fish.
With their pencil-shaped bodies, they are specially adapted for a gravelly stream bed.
They're fascinating in that they can burrow down into the gravel and you know, in that Kauru Kakanui population, they found eggs, I think 25 to 30 centimetres down in the gravel, which is quite impressive.
The spawning season for lowland longjaw is from August through to January, when they lay eggs.
The size of the eggs are quite small. They're about 1.8 to 2 millimetres in size.
The female lays her eggs on rock surfaces, and the male fertilizes the eggs, which develop and hatch.
We're not 100% sure how long they take to hatch, but it'll be a number of weeks, I suspect. Females probably have between 80 to 100 eggs, so they have ability to, you know, produce lots of young as long as they don't have threats.
One of the main threats to lowland longjaw is introduced trout. Brown and rainbow trout from the northern hemisphere were released into the rivers of Aotearoa in the mid to late 19th century for sports fishing. Trout can move through river systems, and when they get to spring-fed streams and tributaries, they feed on native fish like the longjaw and other non-migratory galaxids, causing a dramatic decline in their numbers.
And this site, Fraser Stream, we noticed in 2009, because we'd been monitoring the site for a few years, and we knew there were trout here, but we hadn't done anything about it, or we used to shift them out of it if we could. 2009 we suddenly found that we were only getting a handful of fish. I think we caught 12 longjaws and 25 big nose in the stream, and we went, that's no good. So we put a barrier in. That was our first barrier, and we've since modified it and changed it.
Barriers are structures like weirs built across streams to isolate habitats of non-migratory galaxids and keep them safe from trout. Dean shows me the first barrier set up across Fraser Stream. It's a small construction. A boardwalk goes on top of the barrier. There are small white concrete walls on either side of the stream, which flows down like a mini waterfall over a wooden log.
The height of the barrier is sort of around about 1 metre high. Idea of these barriers is to create a drop that trout can't jump, because trout can jump quite high if they've got a pull to kind of get speed and take off, and they can jump quite high over barriers. But the idea of this process is if you have a fall onto a concrete splash pad, then they can't get any momentum to jump over the top.
Using artificial barriers for conservation is called isolation management.
The term isolation management has been used sort of since the early 2000s, sort of coined originally by American researchers who were using barriers to prevent non-native trout from getting into native trout habitat, and it's sort of a niche use of exclusion barriers.
This is Martha Jolly, a PhD candidate at the University of Canterbury. Martha has spent quite a bit of time out on the Canterbury rivers studying how barriers can help native non-migratory fish.
I usually call it standing in a river thinking about science, which is part of it, but you just feel so privileged to be in these amazing places working with these incredible species.
Martha is a member of Freshwater Ecology Research Group, a team at the University of Canterbury studying and researching river ecosystems.
We really set out to work out whether, exclusion barriers were working for the protection of river resident galaxids. We looked at the barriers that are present at the moment, whether they're working. We put some parameters around barriers. We found that if a barrier was 1.3 metres or higher, it was likely to keep trout out of a headwater stream. And occasionally, if you place a barrier in a stream, you may need to do some trout removal, which is quite similar to sort of fenced reserves in terrestrial conservation, where you might put a fence around a precious species and do sort of predator removal to protect that species.
There are natural barriers that isolate and protect some non-migratory galaxids, like waterfalls that are too high for trout to climb. And when there are no natural barriers, an artificial barrier is built.
Most of our artificial barriers or engineered barriers are in the upper Waitaki catchment, where we've got really vulnerable fish species like Galaxias cubitinus, which is the lowland longjaw, and Galaxias macronasus, which is the big nose galaxids. Some of the other galaxid species like vulgaris and porcispondylis tend to co-occur fairly well with trout, and some of those populations are upstream of barriers, but there don't tend to be built barriers protecting them.
There are many factors to consider when designing an artificial barrier.
There are now sort of 12 barriers actually in the upper Waitaki. It's been a real sort of adaptive management, learning and growing while the barriers have gone in. They all look a little bit different depending on the habitat that they are protecting. One of the problems with that habitat is that it's really low head, it's really low gradient, so you can't put a great big barrier in there. You just end up with a great big back pool that prevents the fish from having good habitat, stops sediment flushing through. So for example, Fraser Spring is a weir, but it has drop logs in it that you can actually remove and reduce the height of the barrier to flush out the system. There's one really close to it at the Waterwheel wetland, which is more of a concrete pad and a culvert with pipes coming out of it. Some of the Otago barriers are actually waterfalls that have been augmented with a concrete weir on top of them. So they all look quite different depending on the system that you're trying to protect, the drop of the gradient of the stream that you're trying to protect. When you can't get the height into a system, we found that if you reduce the pool depth, so put a concrete apron in underneath, that prevented jumping, and also perches, so putting pipes in prevents fish from getting above those barriers.
The Fraser Stream barrier has a special feature to also keep out the native migratory galaxids, whitebait species like the Quaro.
They're called the climbing galaxid, and they're really good at climbing any kind of surface that's a little bit damp and a little bit rough. And Quaro, unfortunately, are quite good at outcompeting the smaller non-migratory galaxids. So, for example, in Fraser Spring, we've actually got a lip on the top of the barrier, our anti-Quaro lip, that stops them from climbing up and round. And there's plenty of space for them downstream. They've got plenty of habitat downstream. A lot of the Quaro populations are now landlocked because of the big dams in the Waitaki further downstream. So they actually use the lakes as their spawning spawning ground and the streams above the lake as their sort of stream habitat. But actually there's plenty of space for them below these non-migratory galaxid habitats.
So the barriers also keep out native migratory galaxids like the koaro, but they have many other rivers to live in and are found almost everywhere across Aotearoa. And the same goes for introduced trout.
Trout are a really valuable part of our freshwater fishery, and they have a huge recreational and social and economic benefit to New Zealand. And nobody is saying that we want to get rid of trout from our waterways at all. What we're finding with the river resident galaxid habitat is that actually it's tiny fragments which it isn't ideal for trout fishery or trout spawning anyway, and the trout that we see in these areas are really quite small bodied. They're not of legal catch limit. There's plenty of room for everybody. There are just some spaces or some environments and habitats where these introduced sports fish are really not helpful and have a very negative impact on our native fish.
Back in the field, Dean checks the last few G-minnow traps.
One to go, I think. Hopefully I've got them all.
He transfers big nose and longjaws found in the traps into buckets of water. If any introduced trout or koaro are found, they will be moved downstream outside the barrier.
Still no undesirables, so that's good news.
Regular monitoring is important, as even with barriers in place, trout can make their way into longjaw habitat, especially during floods that flow over top of the barrier.
One of the worst moments I had here a couple of years ago, I was just walking in to have a look and check the barrier. I do that on a regular basis just to make sure everything's okay, and I was walking up that lower bit and I saw this big trout take off, and I trout kind of sit in the middle of the stream, waiting for food, and when it saw me it just took off, and there was this big bow wave as it swam off, and I just had this absolute horror moment. Oh my God. So we managed to we pulled all the boards out and dropped the level of the stream and got the electric fishing machine. I got some more staff in, and we managed to catch it, and it was over 300 mil long. It was a big fish. So I don't know how long it had been in there, but it had potential to chow down on a lot of these little guys.
Between checking traps, Dean points to a plant growing in the stream, an aquatic weed that's a threat to the longjaws.
So this stuff here is an introduced macrophyte. Monkey musk tends to produce this incredibly fibrous root system that binds up the gravel, and I'll pull some out just to show. And you can see, it's got bits of gravel embedded in that root system. So if this covers the whole stream, effectively it makes it really hard for the fish to burrow down into the gravel, because it's kind of bound up the whole substrate in the stream. You see there's quite large stones embedded in that root mass. And the key thing about it is making sure that you get it before it flowers and seeds and spreads another generation. And this is growing right, you can see actually there's a little spring head here, there's a bit of water seeping out of the bank, so that would be a site that fish would potentially spawn on, so it's just making it really hard for them. Fortunately, one of my staff absolutely loves pulling this stuff out. That makes it a bit easier. This is a native, so it's called a zola, and it floats on the surface, so it doesn't have roots in the ground. So it's not, while it can cover quite large patches of the river, it's not such a problem as that other introduced species.
At Fraser Stream, lowland longjaw numbers have increased from just 12 before the barrier was built in 2009 to 125 in this year's monitoring in March. Big nose galaxids are up from 25 to 270, and Dean did not find any trout or koaro in the G-minnow traps today, so the barriers are working to keep them out. The big nose and lowland longjaws will be counted, measured, and released back into the stream. The fish are all in 2 white buckets, one for those caught in the upper half of the stream and the other for the lower section. This is to make sure they're released in a similar habitat they were captured in. There are more big-nosed galaxias in the buckets than lowland longjaws.
As a rule, for some reason, I'm not sure why it is, but longjaws don't like going into these traps as much as big nose. So even though we know there's roughly the same number of each species in here, when we're using traps, you'll always get a lot more big nose than you do longjaws. What I'll do is do a quick measure. So this is our measuring board.
Dean uses a scoop to carefully transfer a lowland longjaw onto a measuring board, which looks like a small plastic tray with a built-in ruler.
42 mils.
To avoid handling the fish too much, he measures only a few just to get a sample data across different sizes.
So that gives us a size range, and then I'll just do a count.
He scoops the fish, counts them, and transfers them into a bucket for release.
25 and 2, 27. Cool. In that top half we had 71 big nose and 3 long jaws, and as I say, long jaws don't like coming into traps, so that's fairly typical. And in what I call the lower half, 27 big nose and 3 long jaws as well.
The data collected today goes into Department of Conservation records, but similar data collected in a new site will be entered on a form that goes into the National Freshwater Fish Database managed by NIWA.
And then anyone can access that data, and you plot it on a map and see where different species are and all that kind of stuff. It's a really good system.
In addition to using G-minnow traps and electric fishing, eDNA is used to check what's living in the stream. eDNA, or environmental DNA, is genetic material like leaf litter, feces or eggs that plants and animals release into the air, water or soil. So a water sample is collected from the stream, filtered, added with a preservative to keep the DNA intact and sent for analysis.
And it comes back with this amazing spreadsheet of all the different species and relative abundance. It's not absolute, but it gives you a relative idea of how many big nose, how many long jaws, all that kind of stuff. Tells you not just the fish but everything else, the invertebrates, your plant species and all that kind of stuff. And whether there's people that have walked through it, and cows and sheep and everything else, it's quite a cool tool.
Barriers are helping the lowland longjaws for now and will remain part of the solution. But in the long term, other strategies may also be necessary. Here's PhD researcher Martha Jolly again.
Looking forwards, it seems as though interventions such as captive breeding and reintroduction into new habitats is probably going to be required to increase the numbers of these fish in these populations.
I quite like to put some more barriers in and then translocate longjaws to those sites where they don't occur now, and that gives us just a bit more insurance against catastrophic events at some of these other sites that they're in now. And I guess I still hold hope that one day we'll find another population that we didn't know about that's surviving somewhere, but you just can't search everywhere, and that's where our use of eDNA might help in the future.
The lowland longjaw galaxias is not really a household name, so to help with that, a fish celebration day was organized in the region by the Aotearoa Catchment Community Group.
There were representatives from many stakeholders there, so local government, University of Canterbury, ECAN, DOC, also landowners and fish and game, and it's so good for everybody to actually get out onto the river and see the fish.
And it was a great chance for the local community to meet their rarest resident.
It's very cool seeing people light up when they look at these fish and they realise they've got these goofy little faces, but actually they're really precious and really threatened. And they're right here, they're right living among us in these little scraps of wetland that you probably would drive past in the car and you wouldn't even look at. People are fascinated and they want to know more. They're really excited about having these fish in their catchment, in their rohi, on their land. And I think that response is really heartening.
This story was reported and produced by Karthic SS, an independent podcast producer based in Dunedin. Karthic spoke to Dean Nelson, who is the Senior Ranger for Biodiversity at the Department of Conservation in Twizel, and to Martha Jolly, a PhD candidate at Te Whare Wangana o Waitaha, the University of Canterbury. I helped with editing, production help was from Ellen Rikers, and sound engineering was by William Saunders. Learn more and see some photos of the fish at rnz.co.nz/OCW. And if you want to hear more of Karthic's work, you can find the show he makes about Aotearoa's unique wildlife by searching for Tune Into Nature on your favourite podcast app. Tēnā koe I whakarongo mai. Thanks so much for listening. I'm Claire Kincannon. Have a great week. Kia pai te wiki.